While everyone's getting on screen, welcome. Good morning. Thanks for joining us for coffee. Today's program is about the 2-year study that we did with Bigelow Laboratory for Ocean Sciences. It took place in 2024 and 2025. When we designed that study, what we looked at was where is there a place where data is missing, and that it would be really important to have data to complement efforts that others are doing to understand the effects of PFAS. And that is why we designed a study that collected data from sites throughout the lower Casco Bay watershed out to Broad Sound in the Bay. This morning, since we're focusing just on that program and there's a lot to get through, I just want to make sure that that was clear. This morning, to cover that study, I have with me Heather Kenyon, our Science and Policy Associate at Friends of Casco Bay. Heather's role during the study was that she coordinated the entire study from putting together a stakeholder group to help us identify the right sites, to putting together all the data for our field days, which was a super challenge because we had to collect things on the right tides at the right time and be all over the place in Casco Bay. And then Dr. Christoph Aeppli, who's with us from Bigelow Laboratory for Ocean Sciences, he's a real expert in this area, was the lead scientist on this study, put together the study design using the best approved methodologies, both for field collection and laboratory analysis. And then we also have with us a biologist from the Division of Environmental Assessment from Maine's Department of Environmental Protection. That's Jim Stahlnecker. And Jim, his job is to collect fish and shellfish tissues and look at the levels of toxins in them. And so Jim was already doing work to look at toxics in fish and shellfish prior to this study, but he was also collecting samples during the same time as the study. So we wanted to bring you sort of a complete picture of what's in water and then what's in fish and shellfish to make the program good. So today's program, like all the Coffee with the Casco Baykeeper, will be in webinar mode, which means that we'll collect your questions in chat and we'll answer those questions at the end. So chat should appear at the bottom of your screen. And to practice with it, like we always do, my question for you is, name one product that you use that contains PFAS. And for me, I work as a ski instructor on weekends in the winter. The shell that I am assigned to wear as a ski instructor, it contains PFAS. And I have to say, I was doing a full day of training and it was pouring rain, and it was pretty good to have a waterproof coat on. So it's these are such hard questions, hard issues to wrestle with. So that's our question. And this study design, in the process of doing it, and as we were beginning the study, that horrible release of AFFF foam happened at the former Brunswick Naval Air Station. Because that happened just as we were designing the study, just as we were starting the study, we worked with DEP and EPA and added some additional sites to our study. And we got the chance to look at PFAS tracing, the difference between an acute event and a chronic event. And what I mean by that is, like, an acute event is like a heart attack. And so this PFAS spills like a heart attack to the system. And chronic is like you keep letting cholesterol build up in your arteries, and eventually it has a negative health impact. So there's acute and chronic exposures when you're looking at the effects of a toxic chemical in the environment. And maybe that's not the best analogy, but it's an analogy that I hope helps with understanding. So to frame this, Heather, what are PFAS?
PFAS are essentially this class of man-made chemicals and they're characterized by a very specific bond structure and the way that they're made. And that bond structure makes them good for some things that we use, like everybody's putting in their chat, like our rain gear. But that also means that they are very persistent in the environment and they don't degrade. And
It's amazing.
unfortunately, it really is amazing, um, yeah, how persistent they are.
Well, I'm also looking at the answers that people are putting in the chat. They keep popping up, and it's like the range of products they're in. Like I saw toothbrushes. Well, they're in our dental floss. And then someone else
Hmm.
astutely said water. And it's like, oh, I didn't even think about that. I'm thinking about products, but like on a secondary level, it's like it gets into our water. Um,
It does, and it's true. It's truly everywhere.
it's truly everywhere. So, okay, then this may be like a duh kind of question, but why is Friends of Casco Bay concerned about PFAS? And I'm asking you this question in the context of our mission to improve and protect
Right,
the environmental health of Casco Bay and its watershed. Why are we specifically concerned in light of our mission?
right. So we have some fabulous partners that we work with. And their mission is to focus on human health effects. And so we really rely on our partners to take on PFAS and human health. But like you said, our mission is the environment. And so we are concerned about how these chemicals are degrading environmental health and possibly aquatic life. So
Okay,
we're sticking to our job here and worrying about what's going on with PFAS in the environment.
so in other words, is PFAS getting into our waterways? And if so, what impacts is it having in degrading water quality and affecting marine life, which in turn affects human health?
Exactly.
So, given that that's our concern, given that we knew there was sort of a data gap in terms of water quality information, what did we do?
Well, it just so happens we have this research vessel and we've been like collecting water samples for, I don't know, 3 decades or so. So when
Life's
Bigelow,
entire lifespan, basically.
basically, yes. When Bigelow Laboratory reached out to us to partner again on a study, we were really eager to do it. We knew that from our 2023 study that PFAS were present in Casco Bay, and that a larger study would help us dig into that issue a little bit more. So it really is just collecting bottles of water. And I know we have some, yeah, we have some great pictures of all of us in the field. So the sample collection is easy. What was wild was all of the planning and prep that goes into this sample collection because we had to be so careful to not contaminate these samples, given that PFAS is everywhere, like we said at the beginning.
Sunscreen, mascara, maybe Mike and Chris weren't worried about that, but just our clothing,
Our clothing,
our gloves,
Sharpies.
our shoes, everything.
Yes, and Sharpies. We can't we always used Sharpies on the boat. We can't use Sharpies when you're collecting PFAS water samples.
Okay, we'll get back to the study um, in a little bit because we'll hear about the results of the study. But before we get into that, I'd like to invite Jim on screen and talk about the work that some of the work that DEP was doing. We're not going to cover everything. The department's been doing a lot of work to gather information about PFAS levels, but we're going to specifically focus on DEP's SWAT program. Jim, can you explain what that program is? It sounds so cool, like SWAT. You picture you guys out there, dun 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 dun, and you're doing this great investigation. So, what is DEP's SWAT program?
Well, we think it's exciting, but it isn't as exciting as SWAT in the police sense. So, SWAT stands for Surface Water Ambient Toxics Monitoring. It's a program that we operate at DEP. The program was established in 1993. We report back to the legislature. That's how it was designed. The goal is to determine the nature, scope, and severity of toxic contamination in the surface waters and fisheries of the state. It's a statewide program. So I'll show you data from other places as well as Casco Bay, so we can put Casco Bay in context, I hope. We do monitoring theoretically in tissue, sediment, and the water column in all waters of the state. I have a coworker who does freshwater work on contaminants. My role is the estuarine and marine part of the SWAT program, which is, of course, what we're interested in today with Casco Bay. So our PFAS work actually began back in 2013. We had done lots of work on other toxic contaminants, heavy metals, PCBs, pesticides, PAHs in blue mussels, soft-shell clams. So we started there with our PFAS work. And originally, we were only getting lab data for 12 PFAS compounds, and we were getting 2 to 3 nanograms per gram in one particular compound, this PFOS, for a detection level. And to put things in perspective, nanograms per gram is parts per billion. So that's in bumper-to-bumper traffic from Cleveland to the West Coast. That's one car. So it's a very low sort of percent composition or concentration is the word I'm looking for. And then the last bit is these analyses are fairly expensive, so that limits how much work we can do each year. Next slide.
Yeah, so in that regard, I mean, two things come to mind. One is that you're measuring that low, and that's because it's the current understanding in the scientific literature and based on studies is that at such low levels, there could be impacts on human health and aquatic life health. Is that right?
Right. So in that previous slide, I mentioned 2 to 3 parts per billion or nanograms per gram. And our detection limits have gone down an order of magnitude below that. So that's, 10 times lower now or even more. So that's encouraging because, yes, for instance, the human health impacts of PFOS, one of the many thousands of PFAS compounds, can have impacts at relatively low concentrations. We'll talk about something called a fish tissue action level in a little bit, which is one way we look at impact possibly on human health. And the big picture concept I hope I convey as we move through today is that concentrations in shellfish and marine fish, the work that we've done thus far, are quite low and shouldn't be problematic from a human health perspective. Well,
that's reassuring. One of the other things before I ask you about the actual results, and I know everyone's chomping at the bit to get there, but when I asked Heather about PFAS, she indicated there were thousands of PFAS compounds in use, and you mentioned testing for 12, and I know in our study we tested for slightly more than that, but that there isn't yet the ability to test for all of those compounds or to sort of look at the cumulative effect of like more than one compound at a time on marine species. Is that an accurate statement?
Yes, we currently we get 40 compounds. We started with 12 and poorer detection limits, and we're now getting 40 compounds through the EPA method that the lab uses, and we've gotten much better detection levels. So it's a bit of a snapshot or a couple pieces of the mosaic of what's there for different types of PFAS compounds.
Yes. So
but
I guess
we do get a couple of the more important ones from a human health perspective. So yeah.
Sure. So again, we're all working in real time to understand and help evolve the science so that we can have try to make the best responses possible to protect human and aquatic life health. So now, I'm sure what everybody's here for is tell us what you know about PFAS levels in fish and shellfish.
All right. Well, as I mentioned, we started in 2013. We started to do work on some bivalves like blue mussels and softshell clams. Despite doing dozens of sites around the state, the only place that we found the PFOS at detectable levels was in Inner Fore River. So this is the only blue mussel site where we actually detected the PFOS. And these levels are, as you can see, the orange bars are the reporting limit, what the lab can confidently tell us whether there's PFOS present or not. And these levels are just above the reporting limits. So these are, a tenth to 2/10 of a part per billion. So
Okay.
quite low levels. And this is the only place we got anything in shellfish for many years until a little bit of recent work on some clams in the Kennebec. Yeah.
So just quickly before we go on to the next slide, as for our audience, you'll see that during this presentation, there's a focus on this PFOS and PFOA, and that's because these are some of the PFAS compounds that have been studied the longest and been in use the longest. And so that's a lot of what, and they're long chain PFAS, so they don't break down easily. So you'll see that there's a focus on that. And then the second thing I would point out is that the Inner Fore River is not a place where shellfish is harvested from. It's closed to harvesting. So this is not great that there's PFAS in blue mussels, but it's not a place where people are harvesting food sources.
That's correct. It's closed for some other reasons. And there's
Yes.
probably some other contaminants in there based on past work that we did. Probably some bacterial issues as well. So DMR would not open it.
Right. There's CSOs, there's industrial discharge highways. Yeah.
But from a PFOS perspective, this is, at least 10 times lower than any level of concern when we talk about this fish tissue action level. So these levels are low, detected, but very low.
Okay. I'm going to allow you to move on because I know we have a lot to get through and it's just an interesting conversation. Please go on.
All right. Next slide, please. All right. We did some work in American lobster. First, we looked at PFAS concentrations in lobster meat. This again is the PFOS data, a particular PFAS compound. The gray bars here are what we can see, the detection level, reporting level from the lab. And we did composites of 5 lobsters. We did 2 of those composites at each site. And so the level of PFOS is on the left axis, these parts per billion, nanograms per gram. Across the bottom, G West is down on the New Hampshire border, the far left side of the slide, and A East on the far right is up by the New Brunswick border. So I've circled F West and F East, which are Casco Bay. So we can see in lobster meat, there's a barely detectable level of this PFOS in Western Casco Bay, and we failed to detect it in lobster meat in Eastern Casco Bay. And again, in a slide or two, we'll get to compare this to what it might mean for human health. You can see through much of the middle portion of the Maine coast, the PFOS is undetectable, and then we've got, one hit further down east. So we can take a look at the next slide. Several years later, we looked at PFAS compounds, these 40 compounds in the lobster hepatopancreas. This is what a lot of people call the tamale, the kind of nasty green stuff. There's already a no consumption advisory from Maine CDC based on dioxin, furan, and some other historic paper-making chemicals. So you shouldn't be eating the tamale. But we looked at that tissue nevertheless to see what the PFAS levels were like. So we're looking at the same thing to be consistent. This is PFOS. And we can see here the levels in the hepatopancreas were higher than they were in the lobster meat. And again, we can see F West and F East are Western and Eastern Casco Bay. And you can see here the amount detected is much higher than the reporting limits. And so different than in meat and a little bit higher. So let's take a look at the next slide. This just puts both tissues on the same scale. So we see the little short green bars are the concentration in the muscle from 2021, and the blue bars are what we found in the tamale, the hepatopancreas, in 23. We're still with the hepatopancreas below a level that would cause any type of a consumption advisory. And we're not too worried about that because we don't want you to eat hepatopancreas anyway based on the dioxin and PCBs. So let's move on to the next slide. So we did some work in bivalves. We didn't detect a lot of PFAS in bivalves. Blue mussels, soft-shell clams. We did some work in lobster. I started to use some fish. My freshwater counterpart detects PFOS and a lot of PFAS compounds in fish tissue, and we wondered if suspected that they'd be at higher levels in fish than they would be in some of the bivalves and some of the other marine species. This species is a little minnow. It doesn't migrate very far. So to me, it's almost as awesome as a blue mussel because it doesn't move around all over the coast. So we have a sense of where they're picking up some of the contaminants. Next slide. So we looked at Atlantic silverside, the little minnow we just saw in the blue and gray bars, and the top three green bars are actually some harbor pollock, very small pollock that we can catch along rocks right along the coastline. And you can see here the pollock scores were relatively low. The red square shows a site in East Casco Bay called Pond Island, and another site in Casco Bay, some little islands called the Stepping Stones. And these concentrations in fish are pretty low, between 0 and half part per billion. If you move down to the other red square rectangle, these are some silverside scores. You'll note Mare Brook. This is pre-spill, pre-recent spill, I should say. It's not the first spill that occurred at Mare Brook, and certainly there's been a history of training and aircraft firefighting there, which is a source of PFOS. And then you can see towards the bottom of that lower red rectangle, there's the Presumpscot, East End, Fore River, are some levels in silverside that are higher than some of the other background that we see. So we're getting a sense here kind of spatially of where some of the higher, I say higher, relatively higher levels of PFOS are, but these are still quite low.
It's interesting, though, because the Fore River gets a lot of stormwater loading and combined soil overflow loading, and it's a highly industrial area, densely populated. So that looks like an area would be interesting for us to study more and understand better.
And big differences between Eastern Casco Bay and Western Casco Bay, despite Mare Brook. And of course, we'll talk more about the spill as we move on. So.
Yes, I think in fact, it's probably a good time to do that, right? In the course of our studies, we were designing it, this spill occurred. And I do just want to take a minute to say it was a horrible, very upsetting, very frightening event. And it's sort of like, sometimes we have to sort of put aside how we personally feel and focus on the science of it. Like, what do we need to do to understand its impact? And so if we seem very matter of fact when we're discussing it, please know we're all as upset as you were by this spill. But now turning to the fact that we had this acute event while we were looking at all of us studying chronic exposure, tell me what you found as a result of the spill?
All right. Well, the department has response personnel who immediately go out and try to kind of arrest what's spilling and clean up what's there. My role is to try to do some monitoring to see, what's gotten into the environment, particularly into tissues. So we made what we called a phase one follow-up to the spill. We looked at softshell clams and blue mussels and a few American oysters within Harpswell Cove. And Harpswell Cove is immediately adjacent to Mare Brook, which drains a portion of the former Naval Air Station. So much of what spilled, you saw the slide earlier with the foam blowing, it gets into some ponds and streams and drains down Mare Brook towards Harpswell Cove. So we looked at 3 weeks post-spill and then went back another 2 weeks later at 5 weeks. And you can see here on the left, there's some softshell clam and blue mussel data from 3 weeks after the spill. And now we can see much higher concentration of this PFOS in tissues. I was surprised to see how high it got, but the concentrations in the water were extremely high coming from upstream. So it's interesting now we're seeing it readily taken up by shellfish if it's present in the water column. So these levels, initially 25 parts per billion, were above the red dotted line, which is this fish tissue action level I keep talking about. Now, this comes from Maine CDC, Centers for Disease Control. They have some toxicologists that have looked at freshwater fish that are angled and eaten at a rate of 8 ounces once a week. And so when they look at the body size of the average human and how much of this stuff you can take in without worrying about it, they sort of look at how your diet might be apportioned. They've come up with this 3.5 parts per billion, nanograms per gram. And so initially, the clams and the mussels were above that at 3 weeks post-spill. When we went back at 5 weeks, because the water column concentration had dropped down and the shellfish were excreting some of this, the levels had come down, but were still right around this fish tissue action level. Now, okay. I would argue this is still quite a conservative intake level because you can eat this once a week.
So, Jim, we have to get to Chris's presentation too. So I'm going to ask you if you could go through the rest of this information, not so quickly that you don't convey the information, sure, but maybe just get through the rest of what the spill did. Yeah. And I know people are asking questions that will allow for more detail at the end.
Yeah, we can come back and do those questions later. Yeah.
Yep.
All right, next slide. So we went back again later that year in November, and then again the subsequent spring in May. And the good news is that the PFOS levels in the shellfish in Harpswell Cove continued to decline. Marine Resources, DMR, kept the area closed, and we waited until the next spring, and then they opened it the subsequent fall in 2025 after we had the rest of the data in. So here you can see the levels have come down well below that 3.5. The blue bars are the November samples, and you can see it comes down over time by the next May. So little concern as far as intake of those shellfish, which allowed us to get the flats reopened in there. And they're also in this graph, you can see there's some quahogs. Brunswick has a planting program for quahogs, and it was interesting to see that those scored a little bit lower and also came down over time. Next slide. And then one of the other things, we were very, very fortunate, you could call it providence. We were present sampling some of these silverside the day of the spill in Harpswell Cove. So we got a before sample the day of the spill because it hadn't gotten down to salt water and they hadn't taken it up yet. So here we can see pre-spill what the levels were in Harpswell Cove, what we call Mare Brook, which is right up where the stream comes in. And then we did a North Harpswell sample and a Harpswell Center sample much further south and seaward. Then you can see 2 weeks post-spill, we went back, and the levels in the silverside were much higher than they were in the shellfish. Again, the fish seemed to be picking up PFAS more than the shellfish. 4 weeks post-spill, it was coming down. We went back again a year post-spill, and it was way down to around 3.5, even up at Mare Brook, right at the most concentrated site where most of the water that was contaminated came in. And that's about what I have. I just have one slide to sort of talk about.
I guess that's right.
Steps.
The big question I was going to ask you is, what are the next steps for the SWAP program in terms of continuing your work on PFAS?
Well, a lot of this is what we've already got done and have in the pipeline. We have some more work on silversides, spatially, lots of places that we haven't sampled yet to try to get a handle on where there might be some hot spots. Marine Resources has provided us with some small winter flounder and white hake. These are more food fish oriented than the Atlantic silverside, the minnow species. So we'll have some data that I'm working on right now from those species. We also obtained some rock and Jonah crab from Marine Resources. So we'll have some data on these 40 PFAS compounds in crab meat. We're just looking at the meat. So that'll be interesting to compare to the lobster. Because the Kennebec seems to have higher levels of PFAS and specifically PFOS than some other areas, we've done some work on rainbow smelt, collecting some smelt, which are popular food fish and recreational fish. That data is in hand. We collected some American shad, which is another sport fish, an anadromous fish up in the Kennebec. We're looking at both the roe and the meat because they're both consumed by people. And then, of course, we're thinking about what we want to do to maybe go back and revisit Harpswell Cove again, see if it settles down in the long term. And we'll probably revisit lobster tissues again at some point since it's such an important fishery.
Great. Thanks, Jim. I see lots of great questions coming in for you, so I know we'll get back to you a little later in the presentation, but I'd like to invite Christoph on screen now. I got worried for a minute when I saw your background, Chris, and it took a moment for your face to pop up. I'm like, no, he really does exist. Chris was the lead scientist on our study, and this could not have happened without him and the excellent lab facilities at Bigelow. And your analysis, you were really focused on sort of source tracing, understanding where PFAS may have come from based on the type of compounds. Can you explain just really quickly what source tracing means? And then let's just jump right into what lessons you learned from tracing the AFFF spill to loading from the watersheds. And you can even explain what source tracing is as we transition to your slideshow.
Right. Thanks for having me here. So as we just saw, the spill obviously was like an impact in Casco Bay. But when we started the study, the bigger question was what are all the different sources of PFAS that go in and how important are they? And especially when you talk about we want to reduce the load of PFAS in Casco Bay or just in the environment generally, we need to understand how important are these various sources so we know where we can turn the tap off or where it's most efficient. So in Casco Bay, that wasn't really that much known about which sources are how important. And I'll just share like a slide here from Casco Bay that illustrates a little bit the different sources that you might see. If you see the slide, there is a lot of like red triangles. That's all sites where biosolid sludge was applied previously, and that's a known source. Then the black circles are where there is stormwater outfalls or wastewater treatment plants or industrial outfalls, that's another potential source. Then I highlighted in blue arrows some rivers, and rivers are known to integrate PFAS from a lot of areas, like, for example, from agricultural field where PFAS might be, that comes in. So this is another source. So when you look at Casco Bay, there's different regions that may be affected by different sources to various extents. And our question was now, can we identify where are the sources and how important are they?
It's so interesting. I'm going to let you go on, but when you think about Jim's fish tissue samples and then being higher in the Fore River, and then you look at all the sources in the Fore River, it's like, oh, yes, there's some connection there. So please continue.
Right. And this is then why we selected so many stations. So as Heather said in the beginning, this project was very much in collaboration with stakeholders who told us, go to Fore River and take a sample, or Harpswell Cove would be great, or Brunswick, we have this site where there is a stream we suspect to be PFAS. So this is how we came up with the list of 90 stations that we went and sampled and then wanted to do our source tracing. And before I show like some larger source patterns, let me just jump right back into like Harpswell Cove that we just talked about. And we selected 5 sites along Harpswell Cove where we felt like this would be really good to have a temporal monitoring, meaning like just see how is it before the spill, during the spill, and how quickly does it decline again. And this is pretty much mirrors just what Jim just showed, but here just focusing on the water, because the PFAS is in the water and then it can be taken up by organisms. And we measured PFAS in these different sites. And the site 1 shows how the total PFAS level in the water sample goes up after the spill and then goes down subsequently. So this kind of like aligns very nicely with Jim's data showing initially you have an uptake, but then because Harpswell Cove is just a tidal system, it gets flushed out eventually. We did this for all the other stations here, and you see everywhere kind of like the same pattern, goes up, goes down, and the further away you're from the source, the smaller the concentrations are. But you see an impact in all of the stations.
Sure. It's still really interesting, though, because this transect, we were monitoring up to 8 miles away from the spill. And you can see, if you look at the very last bar graph in the lower right hand, that it took 8 weeks, there were the highest levels 8 miles from the spill. So it's just a really interesting look at how long it takes a chemical, a toxin to travel in a marine system.
Right. And the other thing to consider here is that the reason why it declines, it can be like, there can be multiple reasons. We think the most important reason here was just the tidal flushing out. So dilution really made the concentration come down here rather than any degradation. Let me now jump back at the whole bay area, the whole bay results. And what we plotted here was just the total concentration of PFAS at one of the 3 sampling campaigns we did. Do you see the result overview slide?
We see the study area Casco Bay with and then your slide panel on the side.
I'm sorry. All right. Here, is this better?
Yes. Do you want us to see the slide panel on the side, or does it need to go back into?
No. Yeah. Is this here good?
Nope, that I think is probably worse.
Yeah. Okay.
We can just stick with this, given the time. Yeah. Oh, there you go. You got it. Yay.
Sorry. We think like we know how to deal with Zoom by now, but it's still some surprises. So here, basically, my takeaway here is just if you just look at the total PFAS concentration, it doesn't really tell you where the sources are. You can say like Hartfield Cove has bigger circles. That kind of like makes sense because of the spill. In Portland, where it's much more urbanized than like, let's say, kind of like more offshore, the aqueous concentration aren't really that much terribly higher. We have some little streams, maybe around the airport or some landfills that are higher, but it's not like out of proportion. So now, where we go now is saying when we look at the different PFAS levels, our question is, can we almost be like detectives and fingerprint which PFAS comes from the spill, which PFAS is more indicative of like terrestrial sources, and which one is more kind of like marine background? And we did our statistical analysis and came up with some like a blue fingerprint, green fingerprint, and some like a red fingerprint that we can say this is now very typical of these different sources. And on the left-hand side, when you look at these pie charts, it shows you how much these different sources contribute. So, for example, Hartfield Cove is an easy one. The red one is a clear firefighting foam signal. The green one is more like a terrestrial signal, etc. So this study allowed us now to apply these methods and say, like, we can kind of like fingerprint and say how much impact do we have of, for example, the Harpswell spill.
So based on these results, what might you recommend as next steps?
Right. So what this one here shows is now where are the different sources impacting. What it also allows us is to then start making some estimation how important these sources are on a mass base. And I want to jump to a slide here that shows a little bar graph that says, like, if we now estimate how much PFAS gets into Casco Bay, we can make a worst case estimation how much PFAS got into the bay from the spill. This would be the red bar, about 20 kilograms of PFAS. Now we can also look at the rivers, which, as we said in the beginning, are also sources of PFAS because they integrate, and we can multiply the concentration we got by the flow there is and estimate how much PFAS gets into the bay every year. And then you see, depending how big the rivers are, this bar is also bigger or smaller. The biggest river, the Kennebec River, which outflowed and also curves and goes into Casco Bay partially, is almost in the similar order of magnitude as the spill. So this kind of like tells us rivers are the sources, the PFAS that are in the river, they're quite important for how much PFAS gets in there. When you then say, like, well, how can we reduce the load in rivers? You can think of rivers receive PFAS from wastewater treatment plants or agricultural runoffs or just rainwater. And of all these sources, the wastewater treatment plants are the easiest to measure because they have a defined pipe. You can go there and measure. And with some colleagues from DEP, we started doing some estimation how much the load really is. And surprisingly, it's pretty small what the wastewater treatment plants deliver in the rivers. So all the other inputs that go in the river is this agricultural runoff, old landfills, other diffuse sources, groundwater, whatever there is. And this is kind of like the hard part of the PFAS is a lot of the sources are pretty hard to turn off. So that would be kind of like a little a conclusion we draw here.
Yes, thank you. I'm sure there's going to be more questions about this, and I want to bring Heather on to talk about the policy implications of this. But I just want to say in closing, Chris, that this is why we study the whole watershed, and this is why we focus so much on the health of what's the rivers that are loading the estuary, because you can't have a healthy estuary if everything that's loading from land is presenting these challenges to its health. And that's really, really difficult issue to tackle. So thank you so much. I'll bring you back on in a moment for questions. Heather, as we kick off the question portion of this, during the course of our study, like when we were more than halfway through, the Environmental Protection Agency published like these draft criteria for PFOA and PFAS to protect aquatic life, and they published benchmarks for marine water. So can you explain like what's the difference between criteria and benchmarks, just to start, just as quickly as you can?
Sure. I will try to be quick. So essentially, criteria are levels of PFAS or any chemical in water that as long as the level doesn't go above it for a certain period of time, the aquatic life should be safe. Now, as you mentioned, we have these benchmarks. So the criteria, what EPA will do is they'll review all of the data that there is, and they'll decide whether or not there's enough to set criteria. And in the case for freshwater, there were so many studies and so much data that they say that they were like, yes, these are the numbers that we are confident in, that if levels don't go above it, freshwater aquatic life is safe. But there's not enough data in the marine environment. And so they
That's
were
part of
like,
why we did a study. Yep.
exactly, exactly. And so they looked at what data there was, and they were like, there's not enough. We're just going to set benchmarks, which they're not as confident in those numbers as they are in the criteria for freshwater.
Sure. And in fact, for some of the benchmarks we've seen, the numbers actually go down and they get more
Right.
protective when we get to there is enough data to set criteria.
Exactly.
Okay. So Heather, knowing that these came out, did you compare the results that we got to these criteria?
I sure did. So, and just very briefly so that we can get to questions, all of the freshwater samples fell below EPA's acute and chronic criteria for PFOA. There's only criteria for PFOA and PFOS when it comes to freshwater. And for PFOS, all freshwater samples fell below EPA's acute criteria, and all but 6 samples fell below the chronic criteria. And those 6 samples were in Merebrook, which
After the
drains.
spill.
Yep, exactly.
Okay. And remember again what I was talking about between acute and chronic when you're
Yep.
listening to Heather. Anything else that stood out to you from comparing our results?
So we had the benchmarks. So we compared all of our marine samples to the benchmarks, and they were all below, but keeping in mind these are benchmarks and they might change. So we did just, out of curiosity, compare the marine samples to the freshwater criteria, and they also fell below those criteria as well, except for one site at the head of Harpswell Cove, which was, elevated right after the spill.
Yeah, so I think at a very high level, one of the lessons we learned is no more spills. And
yes.
I know we worked hard to support legislation and the efforts of Representative Ankeles and other legislators who were introducing bills to try to help understand where AFFF foam still exists in the state and how to collect and eliminate those sources, and to ensure that what happens at Brunswick Executive Airport now, that the AFFF is removed and replaced with other products. So I do want to make time to get to questions. For those of you who want more, there will be links to two, and Chris and Jim, feel free to come back on screen. There'll be links to the 2 studies that we did in chat. One is a study by Chris that goes into more of the source tracing, and then the other is a paper that Heather and I wrote, sort of goes into more detail about the results that she's talking about and what might be next steps in terms of making sure we have enough information to hopefully someday set water quality criteria for PFAS compounds. Some of the things that concerned us is, you might have noticed on that slide that Heather had up, that they're looking at exposure over a period of time, like 4-day averages or whatever. And we did grab samples, which is typically how you start a scientific study. And then, as many of you know, like, we have switched a lot of our water quality monitoring to continuous data collection over longer periods of time. And that is probably where this study needs to go to. We probably need to take some of these places that are hot spots and figure out how to continuously monitor PFAS levels in the water, and then continue to compare that with the work of scientists such as Jim and others in the private sector who are also helping with this work to kind of better understand the effects of PFAS. And there were some like really, really great questions in the chat that I want to make sure I leave time to get to. So I am going to get to those questions. Give me just a moment so I can get to the question list here.
While Ivy's doing that, I just want to take a moment to thank Bigelow Laboratory for Ocean Sciences for being such a great partner with us, as well as DEP. They're always willing to talk to us, and our funders at EPA, and of course all of our members, because you make our work possible.
There are going to be a lot of questions here, and I don't know. The first question is, how do the PFOS levels we're seeing in Casco Bay compare to levels measured elsewhere in New England and in nearby Canadian waters? And I will say to start this that when Chris and Heather were helping to sort of like spearhead and develop this study, we couldn't find other really comprehensive marine campaigns like this one that collected this much data. So, expert panel, has anything changed since that time, or do we know more about PFAS levels in other embayments in New England? I know New Jersey's done a lot of work, but in our area.
Chris, I'll let you take this one.
Yeah. So first of all, I wouldn't say so. The Casco Bay is really like a comprehensive campaign, and there's not many comprehensive study. There's not many like this. How does it compare in Maine to others? So often people say like, well, Maine, you do so much research, so you must really have a PFAS problem. But the Maine situation is not really that different from other New England states. What we have to say is in Maine, we don't really have former PFAS production sites yet, like you probably have further down south. So we're in this case, we're kind of cleaner than this more industrialized areas. If we go more up north, so we did some sampling all the way up to the Canadian border, there is always a background level of PFAS that's in the ocean just because we have been putting PFAS in the environment for the past 50 years. So wherever you are in the ocean, you'll find some PFAS that's pretty similar to what we found around Casco Bay offshore samples.
So I just wanted, before I go on to more questions, audience, I'm sorry. I tried to move us along, but there's just so much to cover. If my panel is willing to stay on, we'd be willing to stay on an extra 10 minutes to get to your questions. So for those of you who can stay on, Chris, Heather, Jim, is that okay to take another 10 minutes? So for those of you who can, please stay on. We'll get to as many questions as we can, and know that this will be recorded. So if you have to leave, you can listen back into these last 10 minutes. And we have so much we wanted to share with you. So Jim, I'm going to go back to you because of course, seafood is so beloved to Mainers. So there's a lot of questions about that. And one of the questions people asked is, what about PFAS levels in other marine organisms, like the food sources that lobster eat, like what's in lobster bait or in offshore fish or even in seaweed that people consume? Have there been any studies of other fish?
We haven't done a lot of that work yet. Things that you haven't seen today that we have done, we've looked at striped bass within Casco Bay and some fish from the Kennebec River. And the problem with striped bass is they're highly migratory. So you don't really have a sense of where they're taking up some of these contaminants. But those levels were at a threshold where they weren't problem from a human health perspective. There are some other contaminants in striped bass because fish are coming from the Chesapeake and the Hudson that are probably more of an issue. As far as seaweed and some of the lower trophic levels, We haven't done a lot of that work yet. A lot of it has been related to human health or trying to get some spatial patterns. But those are things that we can begin to do in the future. And of course, the other issue we have is we're statewide, so we're having to expend resources in a lot of places and not just Casco Bay. So, yeah.
Yeah, let me just ask you a follow-up question, and then I'll get to questions for Chris as well. I'm going to combine a lot of the questions that I see. First, I know people want to know, like, kind of like how many lobster you test or what's going on. Can you just kind of give an overview when you're looking at monitoring for the entire state, the size of your staff, the cost of all of this, like how much you're actually able to do, and how many, like, for example, how many lobster are you able to test in a year, and how does this program work in terms of, this isn't easy to do?
Yeah, the big limitation typically is the lab cost. We don't do the lab work ourselves. We have to contract it out to an EPA approved lab. And so that's the big bottleneck. So in the case of lobster, we were compositing. All of these samples that you've seen today are composites. We don't test one animal. We're compositing 5, 10. In the case of the silver sides, it's 25 animals in every sample. So we're integrating across a variety, a larger number, and trying to keep the lab costs down. So for the lobsters, we're compositing tissue from 5 lobsters. And in some cases, we've done 2 of those composites just to look at variability at each site. So if we're doing all the way across the state and all the lobster management zones, we try to do like an east and a west, or some of the bigger zones we'll do 3 stations. And then it's, 20 lobsters, 10 from each side, 2 different composites. And so, yeah, the total budget that we typically have to expend for everything in a year, and it's been a lot of PFAS lately, but there are a lot of other contaminants, is about $60,000. And at, around $500 a sample in tissue, it doesn't go very far. So that's why it's been a composite, picking and choosing different things over time and trying to piece it together.
Thanks, Jim. For people who want to know more, could please, there's a link to the SWOT reports, and those are really excellent reports. Like, I actually enjoy reading them, which I can't say for all technical reports. Some I do because I must, but they're really well done reports. Chris, going back to you, there's a question about, do the results after the AFFF spill show a true reduction in PFAS or a shift from the targeted PFAS, in other words, the ones that we know and have methodology to test for, to compounds not captured in the test?
Yeah,
I don't know if you can even answer that, but like, what? That's the question.
Yeah. So that's often kind of the question, do they just disappear into other PFAS? And I have to say, some PFAS can transform into others. And this is kind of like, maybe you heard from like agricultural sites, from like this precursor PFAS that then can go into others. But for the most problematic one, the PFOS, PFOA, they typically stay the same. So if we have a reduction in PFOS or PFOA, that's typically a real reduction. So they're called forever chemicals because they're not really biological active. So for some PFAS, they can transform. This, like most PFAS of concern, I would say that's a true reduction. The question is, why does it reduce? We think it's mostly dilution, maybe partially also sedimenting out, but like most of it is probably just the tidal flushing out of it.
Sure. And I think just to build on that, Jim, it was interesting to look at the difference between like the filter feeder fish and the fin fish. And it looked like what Chris is talking about might bear out in the differences that you saw in levels between finfish and filter feeders, or even just the different kinds of mussels to oysters to clams. Is that accurate?
I think the biggest difference we see is when we jump from the bivalves, the filter feeders, to finfish, we see an increase in the amount of a few of these PFAS compounds that get incorporated in their tissue. Yes. The other thing that we see one of the compounds that Christoph mentioned as sort of having a marine background component, and that's the PFOSA. And we see that more commonly in some of the shellfish than we do the PFOS. And so I wonder if that's linked to, more of the offshore water component, what they're being exposed to. Yeah.
Sure. That makes sense. And then Heather, I also want to follow up with you on this question and what it leads me to, because when we were analyzing the data, you and I, and sort of talking through all this, we were asking ourselves all kinds of questions, like there's only criteria or benchmarks really for, let's say, these two compounds, right? When we're looking at and yet sometimes in the water samples there were many more than that. Like in one water sample, I think in some areas you were seeing like 15 PFAS compounds detected. What concerns you about that in terms of the level of what we're able to study right now versus those numbers?
Yeah, it's a good question. We need more data, right? Yes, there were all of these other compounds in the water samples, but in order for us to really do a complete analysis, we need lots of people collecting more data as to the effects of those compounds. And so that's one of the, as you know, we suggest in our paper that we wrote is that we need more people, organizations conducting studies, so that we can start to understand the effects of all these other types of PFAS that we don't have testing for.
So there's a question for any of you that know the answer to this. Has the phase out of legacy PFAS, and in this case, let's focus on PFOA and PFOS, led to measurable reductions in PFAS levels overall, or are other PFAS compounds filling that gap? Like a brilliant question. And I'm so curious if any of you think you can answer that.
I think one interesting data set is actually from the National Institute of Health, where they measure regularly just this blood serum level of people across the United States for the past 20 years. And this is like really an interesting data set where you see a reduction by about 80% of PFOS and PFOA levels. So it seems that regulation for these compounds and more awareness really led to an outcome. Not that we measured this in this study, but this is just from data I know of.
Jim or Heather, do you have anything to add to that?
I would just add that though PFOA and PFOS, are phased out, we are replacing it. We, as in chemical manufacturers, are replacing it with other types. And that's probably not what we want to do, right? We want to really just shut off the tap on PFAS. And so while these other types are being phased out, we're replacing them. And it's hard, again, back to we need more data, we need more studies. It's hard to keep up with all of these new types of PFAS that are coming out and trying to collect data on them, right?
Yeah. And I think that leads me to another one of the questions that Heather and I were wrestling with when we were working on this paper, which is just this two things. One is bioaccumulation, which some of the questions we're getting at, like, oh, you got bait and then it's consumed by lobster, or, what happens as you get further up the food chain in terms of like that. And then also like if a particular fish or shellfish or human, although we're not the experts on human health, it's like get exposed to 15 compounds, what is the cumulative impact of all of that? And
we don't know.
We don't know.
We don't know.
Chris or Jim, anything to add to we don't know?
Well, to me, it's a twofold problem in that in some of the samples we've taken, shellfish or finfish, we see, in addition to the PFOS or the PFOA, we'll see 5, 6, maybe up to 8 compounds of the 40 that we're testing for. And those are usually in much lower abundance or concentrations. But this is just a snapshot of 40 out of thousands. So yeah, it's definitely the question for the future. Yeah.
So speaking of the future, and we've used up our 10 minutes over. Thank you for those of you who stayed with us. These 2 papers that we've written about the study, we are kind of peer reviewing those right now. We plan to meet with the stakeholders who helped us design the study. We've sent the paper to the Department of Environmental Protection, and the goal will be to sort of talk this over. So there might be some slight modifications to our policy paper as it goes through all of that review, and then to really identify what should we do next. Like we know what we did was a really solid scientific and policy lockstep action, and we learned so much from this, but it's still not enough to fully set like water quality criteria. I can't answer whether there's enough for freshwater yet. Those will come out of the discussions. So there will be further actions and the need for further study. So stay tuned. We will not stop working on this issue. We look forward to continuing to collaborate with the Department of Environmental Protection and Bigelow Laboratory for Ocean Sciences. We're very, very grateful for the funding we received from the Environmental Protection Agency through a National Estuaries Program grant, and from other funders and our members to support this work. And thank you all so much for joining us this morning. Bye, everyone. Enjoy the rest of your day. Thank you, guests.